delighted to, we've got a 10-minute presentation now, and then we have a panel discussion before our coffee break. So please put your hands together for Martin from Acies Bio.
Hi. Hello? Can you hear me well? Yeah? Can you hear me well? Yeah, okay. So we are continuing now with optimism, and as the panelists before said, there is a lot to be done to bring bioproducts on the market, and Acies Bio can help you with that. We have a SmartRoute platform that is dedicated for basically tackling a lot of the key challenges that are needed in order to bring the new sustainable products on the market, thinking about the key technological pillars that are needed to be optimized in order to develop the technology. We talk about microorganisms, so producing efficient strains that are able to grow and produce on the sustainable fermentation feedstocks such as glucose, different side streams, and then combine that with a optimal bioprocess that is also coupled with a efficient downstream process. What we designed in terms of the development process is a platform where we can take all the different steps from starting strain selection, going towards optimization of the production strains using microfluidics and high throughput screening, then finally optimize the strains and optimize the bioprocess and downstream process in a smaller scale, and then go towards the commercial scale where we can execute the production in up to 10,000 liters, and we are expanding that to 30,000 liters in the next year. So we'll be able to not just produce and scale, but also do the flexible contract manufacturing of microbial processes. We can do that at different technology platforms. We are talking here about functional proteins, secondary metabolites, terpenoids, lipids, and also our one carbon bio platform where we can take renewable methanol and do the production of biochemicals using a new sustainable feedstock. We have been around from 2006, working on different aspects, and as I said before, made a lot of investment into the demonstration facility expansion, DSP, so we can really execute a lot of different development projects using microbial technologies. We've done that on a lot of different partnerships, more than 90 partnerships with industrial partners in different final industries such as food and feed, ag bio, pharma. We've done that with industry giants. I can maybe highlight here our partnership with ADM and BASF on production of sustainable surfactants using our C1 platform, but also bridging the gap to some of the portfolio companies that that were discussed previously with VC-backed companies, and doing that really on the different scales. So doing the discovery projects where we take your idea, bring it in the lab, and do the first IP protection of the strains, and then enable the technology to grow towards a pilot scale, producing sample materials, and then bringing it to real industrial scale, and we're really proud of having more than 14 projects where we reached really the production scale across the globe with our partners in the manufacturing. We've done a lot of different projects, but maybe here we are highlighting some of the results that we have been working with, and we are talking about chassis strain development for vitamin production using our SynBio platform of Eurovia. Using highly efficient genetic tools, we developed vitamin production strains and optimized the fermentation to reach high cell density, to reach the KPIs in terms of the strain performance and commercial feasibility. And then we are now scaling this technology with our partner further. For example, we also developed a synthetic biology industrial technology for production of vitamin B, where we, on one hand, introduced the new biosynthetic route into the strain, but then also worked on adaptive laboratory evolution and reached the final titer of more than 30 grams per liter, and also helped our partners with a registration process, so that during the development we were able to make sure that the final registration process will be going smoothly. And then in terms of non-GM based development, we can take the strain, already existing strain that is on the market, and elevate the titers even more, which we did in terms of when we were doing the production of polysaccharide. We took the strain, did the high throughput screening using our adaptive laboratory evolution and microfluidic setting to bring the strain to the next level, producing the highest producing strain on the market. And then using all of these aspects, we can also develop the complete technology, which we did using a non-GM biocontrol agent strain, where we worked both on strain yield increase, elimination of undesired byproducts, and optimization of production media to make the final technology commercially feasible. And this has been done in an integrated way, which led to the final product much quicker. In the scale-up facility, we have worked on, for example, development of nylon precursor. We took Geno strain for caprolactam production, scaled the strain, scaled the process, scaled the downstream, and produced and isolated at our higher scale, produced the first ton of the material in less than 3 months, and basically enabled their project to go further by providing the samples to their partners, and successfully completed one part of the project with our scale-up. So this is really a broadness of what Acies Bio can do. I'll be happy to talk to you if you have your ideas, and if you can think about what and how we can help you in terms of the technology developments, and we can really do a lot of things using our biomanufacturing capabilities in our pilot fermentation, which we are upgrading in the next year, not only to serve as a biomanufacturing, not just as a scale-up, but also as a flexible CMO for your strain, for your processes, and we can do that using different business models. I'll be happy to talk to you about which one is really suitable for your needs. So flexible biomanufacturing, flexible business models in order to bring new biotech solutions on the market. And thank you for your attention.